Welcome to Week 8, Lecture 4 of the course Introductory Animal Physiology, Module Reproductive System, Male. Hi, I am Dr. Tulasi Raman, Assistant Professor, Veterinary Physiology in Department of Veterinary Physiology and Biochemistry, working in Faculty of Veterinary Animal Sciences, Institute of Agricultural Sciences, Banaras Hindu University. In this lecture, we will explore the fascinating topic of the male reproductive system, focusing on its anatomy and functional aspects. Our key objectives are to introduce the male reproductive system and its components, to discuss the structural and functional anatomy of the male reproductive organs, to delve into the process of spermatogenesis, a fundamental aspect of male reproduction. The session is designed to provide you with a comprehensive understanding of the biological process underpinning male reproduction and their physiological significance. I encourage active participation and inquiry to make this learning experience engaging and enriching. Let's embark on this educational journey together to explore the intricacies of male reproductive physiology. So, male reproductive system, it is an analog to the manufacturing complex. So, manufacturing complex means you can imagine it is as a factory, the male reproductive system. So, the primary product produced in this factory is a fertile spermatozoa. That means the spermatozoa which can able to fertilize. Then the hormones and other secretory products that contributes the efficiency of the overall manufacturing and delivery processes. So first is the what is the primary product. Next, to enhance the manufacturing and the delivery process, the hormones and the other secretory products regulating it. Then what is the primary sex organ? That is the manufacturing unit and the assembly plant for spermatozoa is that testis, which is a primary sex organ where the primary products, that is fertile spermatozoa, is produced. Then the finishing section, that means once the spermatozoa is produced, a specialized cooling mechanism is required for that production, right? That is called as Pampniform plexus, where the counter current exchange mechanisms and all will take place, that we'll see in the upcoming slides. So with the help of that, the spermatogenesis process will take place. Once the spermatozoa is formed, that will pass to the retitubules and then it reaches the efferent ducts, from where it enters the head part of the epididymis. From it will move to the body, that is the corpus of the epididymis. So the finishing section of a spermatozoa, that means the primary product is that in the epididymis. So first it will go to the head, from head it will go to the body. Now you know the epididymis is having three parts. One is head, body, and as well as the caput. Caput, corpus, and then cauda, that is the tail portion of the epididymis. Now once the spermatozoa is produced and reach the epididymis, where overall manufacturing, I told the hormones as well as the other secretory products, that will make some changes in the spermatozoa and then it will make it fertile. And these changes will take place in the head and body portion of the epididymis. From there, a gradual transport of spermatozoa will take place that ultimately reaches the cauda portion of the epididymis, which is nothing but a storage organ. The storage organ of a fertile spermatozoa with semen. So that means the seminal plasma, that is called as warehouse or the shipping center. So now what we have seen, it is a factory. So in the factory, the primary target, I mean the product, is that fertile spermatozoa. How this fertile spermatozoa is produced? By a process called spermatogenesis. For the spermatogenesis, where it will take place, it takes place in the testis, that is the manufacturing unit and the assembly unit. Once the spermatogenesis, through the spermatogenesis process, the spermatozoa is formed, then it will reach the finishing section such as the caput as well as the corpus of the epididymis. Then from there it will gradually reach the tail or the cauda portion of the epididymis and then get stored. That is a warehouse or the shipping center of the semen. Then once in the spermatozoa in the tail of epididymis, that is having a capability to fertilize, and this spermatozoa is motile, whereas the earlier one, which is non-motile, that means it can able to swim. Then upon sexual excitation, The spermatozoa in the tail, that is the chorda portion of the epididymis, that is shipped via contraction of the epididymal duct and ductus deferens. So through these contractions, the spermatozoa, which is motile, it will move to the new location, that is new center, that is called as pelvic urethra. Then in the pelvic urethra, where the final alteration of the product will takes place, and ultimately it is packed. Once it is packed, then during emission process, that is, it is mixed with the fluids from the accessory sex glands, and collectively this mixture of fluids is called as seminal plasma. Okay, so when you say semen, it contains spermatozoa and then seminal plasma. Okay, this is called as a semen. So ultimately it reaches the final alteration and the packaging is done and reached the pelvic urethra. Now once the sperms reach the pelvic urethra, that means it is mixed with the seminal plasma from the accessory sex glands from the fluids, then it is available for delivery. How it will be delivered, the spermatozoa, that means the semen, it is delivered by ejaculation process. Now in this, the delivery system is the penis as well as some specific muscles which surrounds the penis, that is responsible for the erection and protrusion of the penis and the ejaculation of the semen. These are the process which is involved for the delivery of a spermatozoa in the female tract. Then let us come to this is about the simple introduction about the overview of a male reproductive system, how it is produced and it is getting delivered, that's all. Now the male reproductive system, what it comprised of or what it is made up of. The male reproductive system is made up of the primary sex organ. The primary sex organ is what? Testis. Okay, that is the primary sex organ. Then the next is that excurrent tract. So this excurrent tract is made up of retitestis, which is continued with the efferent ducts, is also called as vasoefferentia, and epididymis, then vas deferens, and then urethra. These all structures which is called as a excurrent tract. So from the testis, it will be continued as a retitestis. From retitestis, it will continued as efferent ducts or vas efferentia. Then it will reach the epididymis, the three parts. Then epididymis to vas deferens, and then ultimately it reaches the urethra. Now in this process, you will have the next important structure, that is compound called as accessory sex glands. So the accessory sex glands is made up of ampulla, seminal vesicles is also called as vesicular glands, then prostate and bulbourethral or Cowper's glands. So these glands are called as the accessory sex glands, which is responsible for the secretion of the seminal plasma. And the last is that delivery structures such as penis and then prepuce. Prepuce is the structure which covers the penis. Now first we will see the primary sex organ, that is testis. So the testis is usually a paired structure in almost all the animals and situated outside the abdomen in most of the species. Whereas in some species it is even present inside the abdomen, right? And wherever it is present outside this abdomen, it is situated in a purse-like structure called as a scrotum. So it will be like this. So this is the scrotum. Inside you will have the testis, right? So this is a scrotum, outer structure. This is a purse-like structure or sac-like structure. Inside that the scrotum is present. Now this testis is hanged, or you can say suspended within the scrotal sac with the help of a spermatic cord. Okay, otherwise it will fall, but it is hanging, or you can say it is suspended in that sac. So it with the help of the spermatic cord. So the spermatic cord attached proximally and help to suspend the testis within the scrotal sac. Then distal end of the spermatic cord is attached with the scrotal ligament. So the spermatic cord had as a proximal end as well as distal end. The proximal end is attached with the testis to suspend the testis within the scrotal sac, and the distal end is attached with the scrotal ligament. Then the species such as mice and bat, where the testis is just lie inside the body, okay, during the non-breeding season. Whereas during the breeding season, what will happen? The testis will try to come out in the species such as mice as well as the bat. Whereas the species such as rat, rabbit and camel, the testis situated in the perineal region, okay. And then the species such as cat, tiger, lion, etc., that is a feline species, you can say, these all species where the testis is located at the subanal region. Sub means below, right, on the sides of the anal region, you can say. Then species such as monotremes, armadillos, sloth and elephant, rhinoceros and avians are the birds where the testis is located in the intra-abdominal, that means inside the abdominal cavity. Then coming to the seasonal breeders such as stallion, ram and camel. In these species, depending on the season, when it is approaching the breeding season, then that time the size of the testis increases. Otherwise, the testicular size is always remains small in these species. So, the testicular size varies throughout the year in case of a seasonal breeders, but this is not the case with the other breeders. Then next will be the testis structure. Testis is composed of three structures or three parts, you can say. The first is the testicular capsule, then second is that mediastinum, and the third is parenchyma, or you can say seminiferous tubule. The testicular capsule, what is meant by capsule? Capsule means it always surrounds something, it encompasses something. Okay, so here also, as I said, it is a testicular capsule, that means it is just encapsulated the testis. Okay, so now this encapsulation is done by 4 layers. Okay, of course, I am not going in detail of the 4 layers. Let us come to the next structure, that is a mediastinum. This mediastinum is divided into lobules, okay, by trabeculae. Okay, so if you see the structure of a testis, then it will come like this, right? Now these are the seminiferous tubules. So this is a trabeculae, you can say, right? These are the trabeculae. So the mediastinum, it is divided into lobules. These trabeculae making the seminiferous or the structure into a lobule structure, right? So that is a mediastinum. Then third is that parenchyma, or you can say testicular parenchyma. This is comprised of seminiferous tubules, which is lined by different generation of maturing germ cells and Sertoli cells. Okay, so what is that? The testicular parenchyma is comprised of seminiferous tubules. These seminiferous tubules consist of the different generation of the germ cells, okay, such as spermatogonia, starting from, it is a basal part and then adluminal part. Adluminal part means it is facing towards the lumen, that is called as adluminal part. So from the basal side, if you see the germ cells, first it starts with the spermatogonia, then it will go to spermatocyte and then spermatid, and the last one is spermatozoa. Okay, so spermatozoa is towards the lumen, but spermatogonia, which is present at the basal part of the seminiferous tubule, basal compartment. Then the seminiferous tubules have different cells. Okay, it is having different cells. So first cell is called as testicular cell. So these testicular cells are nothing but the Leydig cell. Okay, then you can say the interstitial cell. It is also called as interstitial cells. Now why it is called as interstitial cells? Because it is present in between, okay, the space between the tubules. That is why it is called as a interstitial space. The other name is name of the Leydig cells. So Leydig cell secretes the LH, but in males it is called as ICSH, not LH. ICSH means interstitial cell stimulating hormone. So Leydig cell, another name is interstitial cell. So the interstitial cell secreting some stimulating hormone, that is why it is called as ICSH, interstitial cell stimulating hormone. That is another name of luteinizing hormone, right? In males it is called as ICSH, in females it is called as luteinizing hormone, right? Because here there is no luteal cells, right? So what is the function of this LH? It mostly the Leydig cells controls the activities, okay, activities of the seminiferous tubules. Then next important testicular cell in the seminiferous tubule is the germinal epithelium, okay, germinal epithelium. Now this germinal epithelium is responsible for the production of spermatozoa. As I said, germ means germ cells, right? Germ cells means what? Where the spermatogenesis will takes place, which contains the germ cells. That means the spermatogenesis process will takes place here. Now the third important cell in the seminiferous tubule is that is the testicular cell, is that the Sertoli cells, okay. The Sertoli cells is also called as sustentacular cells, okay. Now the Sertoli cells or sustentacular cells numbers remain constant, okay, or remain constant even after puberty, okay. But in seasonal breeders, the volume as well as the function increases during breeding. Even the testicular size also, I told you that its size varies with the breeding season. So occurs similarly in the Sertoli cells also, the numbers as well as fluids or the components which is secreted by the Sertoli cells also increases or varies with in the seasonal breeders. Now let us see the functions of the sustentacular cells or the Sertoli cells. It acts as a nurse cell. The most important function of the Sertoli cell is it acts as a nurse cell. So nurse, when we say, that means what? It tries to provide the nutrient to the germ cells which is required for the spermatogenesis to take place, and then this spermatozoa and aids spermatogenesis by secreting the SCF. SCF means it is stem cell factor, okay. It is stem cell factors. So by secreting this, it provides nutrients to the germ cells as well as the spermatozoa that helps in the spermatogenesis process. That is why it is called as a nurse cells. Then it regulates the spermatogenesis. How it is regulating the spermatogenesis? By releasing the stem cell factors. And then it also participates in the blood-testis barrier. Blood-testis barrier, okay. So how it is forming the blood-testis barrier, we'll see in the next slide, okay. I'll tell you. And then the Sertoli cells secretes the ABP. ABP means androgen binding protein. So the testosterone, when it gets secreted, it will just go and bind with this protein, right? So the testosterone binds with the ABP that helps or aids in the epididymal transit as well as the maturation of the spermatozoa. So this is very, very important. Then only the testosterone can act on the spermatozoa where it gets matures. And then the Sertoli cells also secretes an enzyme called as 5-alpha reductase. This 5-alpha reductase is essential for the conversion of the testosterone from inactive form to active form. So inactive form here is that testosterone, whereas the active form of testosterone is that 5-alpha dihydrotestosterone, that is DHT. This is the active form of testosterone. For that it requires an enzyme 5-alpha reductase. Then Sertoli cells phagocytose the defective germ cells, and then it produces the activin, inhibin, anti-Mullerian hormone and insulin-like growth factor 1. Then it also helps in the maturation and proliferation of the Sertoli controlled by the FSH primarily. So this Sertoli cells function, whatever we have seen so far, these are mainly regulated or controlled by the follicle stimulating hormone, or you can say FSH. Then the Leydig cells. The Leydig cells is also called as interstitial cells, already we have seen, and what it does, the main function is to secrete androgen or the testosterone. Now, I told you about the blood-testis barrier. Now let us see what is blood-testis barrier. Blood-testis barrier is nothing but it is a barrier formed between the vascular endothelium of capillary blood vessels and then epithelium of the seminiferous tubules. Remember, when we say of barrier, that means it is in terms of one is endothelium, another one is epithelium. Even in blood-brain barrier and the blood-CSF barrier also, these two components will come. Similarly, here blood-testis barrier, where the vascular endothelium means it is the capillary blood vessels, and then epithelium where it is present in the seminiferous tubule. So a barrier is formed between them. Why it is required and how it is made? It is made by the tight junctions, formed by the tight junctions. These tight junctions are present between the adjacent Sertoli cells, which forms a barrier that divides the seminiferous tubules into basal and then adluminal compartment. Okay, so this is the part where divides the seminiferous tubules into basal compartment and adluminal compartment. Now, why it is required? Because it need to isolate the germ cells from the circulatory system and then as well as the lymphatic system. So, why it is required? It causes the immune suppression by considering germ cells or the spermatozoa as an antigen, okay, and then it produces the anti-sperm antibodies that leads to the infertility in the humans or even in the animals, okay. So just to avoid this infertility, the nature have created the blood testis barrier where the Sertoli cells will come and then it just present adjacent to them. With the help of the tight junctions, it divides the seminiferous tubules into basal compartment and adluminal compartment. Then which structures are involved here? The vascular endothelium as well as the epithelium of the seminiferous tubules. So this is required to protect the spermatozoa from the circulatory as well as the lymphatic system to avoid the production of the anti-sperm antibodies in the circulatory system. So spermatic cord next. Spermatic cord, already I told you, it is a structure which just hang or you can suspend the testis into the scrotal sac, right? So it is a tubular structure that suspends the testis into the scrotum or the scrotal sac. So related with that spermatic cord is the descent of the testis. So descent of the testis is nothing but it is a migration of the testis from the intra-abdominal location to the scrotal sac. Okay, this is called as testicular descent. Now, how this testicular descent will take place? There is 3 stages are there. First stage is called as nephric displacement. In the nephric displacement, what it happens? It causes the detachment of the gonads from the mesonephros. Okay, so this will take place in nephric displacement, and then this after the nephric displacement, once the gonads is detached from the mesonephros, then the Leydig cells releases the INSF3, that is androgen independent insulin like factor 3. Okay, so that is INSF3. So Leydig cell produces androgen independent insulin like factor 3, and this INSF3 causes the swelling of the gubernaculum. So gubernaculum is nothing but the myofibrous structure, okay, where the testis to scrotum it is present. Okay, the structure is present in the testis to scrotum where the myofibrous structure it is, where the swelling will take place of the gubernaculum that results into the initiate the testicular descent. But in order to initiate or favors this testicular descent, it also requires the increased intra-abdominal pressure. So 2 factors are involving. One is that swelling of the gubernaculum, and then the gubernaculum is nothing but a canal, right? So swelling will take place. As a result, the intra-abdominal pressure, these 2 factors favors the descent of the testis. So that it dilates the inguinal canal. The gubernaculum swelling as well as the increased intra-abdominal pressure dilates the inguinal canal. As a result, the testicle migrates. Okay, so the hormones such as estrogen that prevents the swelling of the gubernaculum. As a result, what will happen? Definitely the testicles will not descend. And then whereas in the epidermal growth factor, okay, which is secreted by the testis, where that will causes a positive effect on the human chorionic gonadotrophin release from the placenta, and this placenta will act on the Leydig cells to release the androgen. Okay, now This androgen causes the swelling of the gubernaculum, whereas estrogen, it prevents the swelling of the gubernaculum, that you need to remember. Then the disorders which is related with the descent of the testis, one is that cryptorchidism, that is failed to descent of the testis. So you can say unilateral or bilateral cryptorchidism. And then next is that inguinal hernia. So inguinal hernia means when the intestine drops into the scrotum, or intestinal enters into the scrotum, right? So this is because of the enlargement of the inguinal canal. So as the inguinal canal enlarges, that means it widens, dilates, as a result the intestine will enter into the scrotum, right? So this is mostly common in the stallion species, right? And cryptorchidism also, it is quite common. The incidences are higher in the horses when compared with the pigs, okay? Then coming to the third aspect of the testis, that is called as thermoregulation of the testis. So in the thermoregulation of the testis, mostly it is required. Why it is required? Then only the spermatogenesis to enhance the thermoregulation is very, very important. Now how it helps the spermatogenesis process? By decreasing the temperature of the scrotum around 2 to 6 degrees Celsius from the core body temperature. Now how it is achieving this 2 to 6 degree Celsius difference from the core body temperature? For that, there are 5 anatomical features are helping. The first one is called as tunica dorsi smooth muscle, okay? Now this controls the scrotal skin. Now how it controls the scrotal skin? This causes the relaxation and contraction of the scrotal skin. When it relaxes, what happens? It increases the surface area. So by increases its surface area, it is trying to help in the transfer of the heat load, okay? The next is the cremaster muscle, that is with the help by elevating and distension of the testis. That means the testis, the scrotum, either it will come closer to the body or it will go away from the body. So during winter season, you can say the testis used to be located closer to the body because of the chill temperature outside in the environment, whereas in the summer it used to move away from the body because of the higher environmental temperature, or you can say higher temperature of the body, right? Then the third important mechanism is that countercurrent heat exchange mechanism. That means countercurrent means what? One it will go and one it will come in this way, opposite way. So the veins which is moving away from the testis, that will carry cool blood, whereas blood, the artery which carries blood towards the testis carries a hot blood. So as it is moving in a spiral form, that is called as Pampiniform plexus, okay? So in this structure will enable the transfer of heat from the artery to the venous. As a result, the lesser temperature blood will reach the testis with the help of the Pampiniform plexus structure by the countercurrent heat exchange mechanism system. Then the fourth way is that absence of the subcutaneous fat layer in the scrotum, right? Due to that reason, it is easily dissipates, otherwise it will act as an insulating layer. Then the abundance of the sweat glands, and the last is that this abundance of the sweat glands is required, which is on the scrotal skin, promotes the evaporative heat loss. Then in RAM, one special feature is called as summer sterility. So in summer, what happens? Usually in the RAM, the artery as well as the veins which is present superficial to the scrotum, okay? So during the hot and humid conditions, what happens? It affects the thermoregulation process. That means the heat dissipation. So as a result, it causes the heat exchange mechanism will be lost. So due to that reason, it causes a temporary sterility in the summer season. That is called a summer sterility. And then in avian testis, if you see, that is elliptical in shape and present usually in the top of kidney. Now, as you know that body temperature is quite higher and mostly the testis is present inside the body, intra-abdominal. So how it is maintaining the spermatogenesis, or it is forming the spermatozoa by 3 important mechanisms. One is that the testis is just located adjacent to the air sac. So due to this reason, the thermogenesis is very effective. It is transferring the heat load. And then second is that mostly spermatogenesis will takes place in the night time, where that is the coolest part of the day. And the third is that the heat resistant, all the spermatozoa of the avians are heat resistant. So by this way, the spermatozoa is produced in the birds. Now, let us come to the excurrent tract. That is, once the testis is over, the next structure is retitestis, right? So the retitestis is nothing but it's a collecting reservoir of a spermatozoa. And then next is the efferent ducts, where it connects the retitestis with the epididymis. Then epididymis, there are three parts. One is head or the caput, body or the corpus, tail or the cauda. Now, it is having four cells. The first cell is the principal cell, where it helps in the or participates in the blood epididymis barrier. The next is that basal cell. It is nothing but the macrophage of the male reproductive system. And then the clear cells, that means it trying to clear the cytoplasmic droplets. Once the spermatozoa is formed, whatever the leftover cytoplasmic droplets are there, it will try to clear. Then the halo cells, that is required for the immune protection. Okay. And next structure is the vas deferens or the ductus deferens, where it is nothing but a temporary sperm storage before ejaculation. And the terminal part form a sac-like structure is called as an ampulla, okay, which is an accessory sex gland. And next structure is that urethra. Urethra, it carries of the urine from bladder as well as semen from the excurrent tract. Now, there are two segments in the urethra. One is pelvic urethra, another one is penile urethra. So, the pelvic urethra, it is from the pre-prostatic as well as prostatic. There are two types are there. I mean, two segments you can say. This pelvic urethra again further divided into pre-prostatic and then prostatic. Pre-prostatic means it is from the vas deferens to the and then up to the prostate. And then the prostatic means it is from the prostate to the penile urethra, right? And then penile urethra is nothing but it is the terminal part of the penis where it excretes, right? Then the erection process. It is related with the ancillary organ, right? That is the penis. The erection is nothing but a psychosomatic event, okay, where there are two important muscles are involved in it. One is ischiocavernosus muscle and another one is called as corpus cavernosum and corpus spongiosum. These are that is the corpus muscles, okay. One is ischio-cavernous muscle and then corpus muscle. The corpus muscle means corpus cavernosum and then corpus spongiosum. So, it causes the contraction of the ischio-cavernosus muscle. Once it causes the contract, as a result, the venous outflow is blocked. So, as a result, it is getting accumulated. Now, this leads to the parasympathetic mediated relaxation of the corpus cavernosum and then corpus spongiosum muscle. So, this leads to creates a space, okay, that this space is called as cavernous space. And in this cavernous space, the blood is present. So, it is engorged with the blood. And as a result, the penis is elongated and then turgid. Of course, I have not entered into the nitrous oxide based mechanism. So, here it is a simple erection process. It is just by creating a cavernous process spaces where the blood is oozed into it. As a result, it leads to the engorgement. That is the penis elongated as well as and turgid. Now, next will be the birds, the accessory sex glands. Now, the birds do not have penis like mammals, and instead of that, it is having a papilla. Papilla is nothing but a small erectile tissue, and these penis are developed copulatory organs. The birds develop the copulatory organ like penis that is present in swan, duck, geese, and ostriches. In these species, you will find the penis like structure, whereas in others, you will find only the papilla. Now, what is the function of the papilla? Mostly it is used to identify in the day-old chick to differentiate whether it is a male or female, right? And then mostly birds lacks the accessory sex glands. Now, the next portion is that accessory sex glands. Here you can see the position of a testis in different animals, right? So, in bull you can see here, this is the penis, right? And here in ram, this is the penis part, and this is the stallion, here you can see, and then this is the boar, that is pig. You can see where the testis is there, and this is the penis part. So, these are the different species. You can see the location of the sperm, the testis. Coming to the accessory sex glands, the accessory sex glands causes the ejaculate. Okay, what is this ejaculate? Ejaculate contains spermatozoa as well as the semen. Now, there's accessory sex gland. First one is the ampulla that is found in ruminants as well as the stallion. Next is the seminal vesicles. Mostly it is located near the neck of the bladder, where in bull, ram, buck, stallion and boar it is present, whereas the tom and the dog it is absent, the seminal vesicle. And then prostate gland, it is present in all domestic males, but in some many books you used to find that in dog the one and only accessory sex gland present is that prostate. Okay. The bulbourethral gland or the corpus gland, this is large as prostate in tom. Okay, see here in tom only, but absent in dog. And then in llama, alpaca and camel, it lacks the seminal vesicles, but bulbourethral and prostate are present in the llama, alpaca and the camel species. Coming to the emission, emission is nothing but the release of spermatozoa and the accessory sex gland fluids into the pelvic urethra. This process is called as emission. Next is that ejaculation. Ejaculation means what? Forceful expulsion of the semen from the urethra. So that is called as ejaculation. Then after ejaculation, what will happen? The penis will withdraw into the prepuce by with the help of the contraction of the retractor penis muscle. Due to this reason, the penis will be retracted into the prepuce. So emission, ejaculation, after ejaculation, what will happen to the ancillary organs we have seen. Then coming to the spermatogenesis process. What is meant by spermatogenesis? Spermatogenesis is a lengthy orchestrated process. Okay, orchestrated process means what? It is a one by one it will do, and a downstream process. So it is a well orchestrated process, lengthy orchestrated process, where the diploid stem cells, diploid means 2n, right? Diploid stem cells which is present at the base of the seminiferous tubules, where the spermatogonia is present, right? That divide through the mitotic division, right? To maintain their own numbers. Own numbers means it is 2n in number. And then whatever the cells are formed from this mitotic division, that will cyclically produce the progeny. So progeny means some daughter cells. So this daughter cells will undergo further meiotic division, either first meiotic and the second meiotic division, and that get differentiated into the haploid spermatids. See, from the spermatogonia, then it became a progeny cells, and then progeny will enter into the meiotic division, and ultimately it forms a haploid spermatids. So diploid become haploid, and this spermatids released as a spermatozoa by a process called spermiation. So in today's class, what we have seen is that the introduction about the male reproductive system, that means starting from the what is a primary sex organ, then what is excurrent tract, and excurrent tract means what are the different components present in the excurrent tract parts, then the different accessory sex glands, then the ancillary organ and its function, then spermatogenesis definition we have seen. Okay, so with this today's topic I am ending. Thank you.